So welcome everyone. It's a pleasure that you all are joining this webinar. We are at 10 o'clock here local time, so it's time to start. Thanks for joining again. And before I go into the topic, just quickly some housekeeping. You have seen that this webinar is recorded. So just in case that you know this, but as a participant, you are not visible in the webinar anyway. So welcome to the final event basically of the microservices project. The microservices project is, as the title says, about predicting climate change impacts on the crop microbiome and the cascading effects on ecosystem services delivery in agroecosystems. This is a Biodiversa funded project, so supported through the different national funding bodies from Switzerland, France, Spain, Germany, and Greece. And so my name is Martin Hartmann. I'm working at ETH Zurich, and I am the coordinator of this particular project. And I will basically do a quick introduction here and then guide you a bit through the webinar. But basically, the ones that speak about the research are other people that I would like to quickly introduce here. So these are Dimitrios Gormboudakis from the National Observatory of Athens in Greece, Pablo Sanchez-Cueto from LEITAT in Spain, Elena Kost from ETH Zurich in Switzerland, Arifina Pintarti from INRAE in France, and Rainer Jürgensen from University of Kassel in Germany. So before I come to the agenda, no, actually I can't go to the agenda. Sorry about this. So I will do a very, very brief introduction to present the actual goals of the microservices project. And then we will have two main parts where we present what we have done in these last 4 years. Part one is European-wide, about the climate change effects on soil biodiversity and also soil functionality. So we have with Dimitris and Pablo 2 speakers that will talk mostly about this part of microservices. And then we have a second part, which is more about how different cropping systems, so organic and conventional cropping systems, might modulate the response of the microbiome to a climate change stressor, which is in this case drought. And then we will close the session with a Q&A part and closing remarks. And speaking of the Q&A, you have the Q&A box where you can enter your questions basically during the entire webinar. So please do so. We will take all the questions at the very end, so we will not stop after each talk to answer questions, but we will compile questions that I will then redirect to our speakers. The questions that might be very easy to answer, I can answer them already by writing into the Q&A box. So please make use of that Q&A box and write your questions if you have any during the different presentations. So with this, I would like just to give a brief introduction to the whole topic and the project and the project goals. I think this is how agronomists have looked at plants for a long time, basically growing in a substrate that's called soil, basically filled with a bit of nutrients and giving basically structure to the roots, but treating the biology more or less as a black box for a long time. Nowadays, we know soil is full of life. So we know that around 1 teaspoon of soil harbors billions of organisms, so with thousands to 10,000s of different species. And these organisms fulfill very, very important functions in an agricultural system, for example, or in a cropping system. On the one hand, obviously, they are important for nutrient cycling, here depicted as the nitrogen cycle, for example, starting with nitrogen fixation, nitrification, and then denitrification, and providing these nutrients in the end to the plants. And they do this not to a small degree. Just as an example, we estimate that the contribution of microbes to nitrogen fixation is around 100 million tons per year. So this is pretty much the same amount as we apply synthetic fertilizer to our agriculture fields globally. But you also see that under certain conditions, with the arrow up, there's also certain greenhouse gases that can be produced, for example, nitrous oxide in the case of the nitrogen cycle. And that brings me to the second function, that microbes are also very important for climate regulation because they balance emission and consumption of greenhouse gases. A good example is the methane cycle, where we have the methanotrophs on the right that consume actually atmospheric methane, and the methanogens on the left that actually produce methane, and they also do this to a very large degree. So basically coming up to around 70%, for example, of the methane that's produced is coming from microbial processes. But also, for example, 60% of the nitrous oxide that is produced in agriculture system is microbially done. So these are large amounts of greenhouse gases, and therefore the climate regulation by these microbes is an important contribution globally. Microbes are also very important for modulating the stress tolerance of plants to abiotic stresses, such as, for example, drought or heat or flooding and so forth. For example, we have mycorrhizal fungi that help the plant to take up nutrients, of course, but also water, for example, by inducing aquaporins, water channels into the root. We have different kinds of microbes, like depicted here as Aspergillum and Arthropacter, that are producing different kinds of plant hormones that start modulating the growth and development of roots and of the plants, and therefore also modulate basically the response of the plant to stressors. These stressors can be abiotic, but they can also be biotic, so they're also very important for disease and pest control in the end. Bacillus thuringiensis, for example, we know is a powerful organism to kill off actually pests, or also the fungus Beauveria bassiana that can kill off a certain kind of insect pests like the May beetle depicted here. And last but not least, microbes are enzymatic powerhouses, basically to degrade all kinds of substances that are in the environment, and therefore help us actually also to bioremediate the environment, for example, to degrade plastics like phenol rings in the terms of Aspergillus, or to also degrade pesticides and pharmaceuticals that are accumulating in the environment. So this is just a very short summary of very important functions that microbiomes basically provide to agroecosystems, and therefore we need to make sure that microbial diversity and its functionality is actually maintained or enhanced to make sure that we can produce food sustainably. And so one threat to microbial diversity and functions is climate change. And this is where microservices comes in. Microservices is all about climate change effects on microbiomes, on crop microbiomes, basically with a major focus in this case on wheat. And so our first objective was indeed more on the climate side. Can we use regional climate models to predict future shifts in our agroclimatic zones, in our climatic conditions of agroecosystems across Europe? And basically doing this using high-resolution regional climate models. And Dimitris will talk more about what they have done at the National Observatory of Athens to basically predict how, in the future, these distribution of agroclimatic zones might look like. Once we have basically the knowledge of how climate would look like in the future, and we can basically assess biodiversity at the moment, and we can assess the climate at the moment, we had as a goal to characterize shifts in soil biodiversity and multifunctionality along these agroclimatic zones, basically using data from the Lucas Soil Network, but also Earth observation data from Sentinel-2. So there also Pablo and Dimitris will update you on what we have done in this case. And with this information in the end, we wanted to forecast, right, to predict basically, okay, under these future climate conditions that we have modeled, and knowing the current climatic conditions and the current state of the biodiversity, how would soil biodiversity and its functions look like under future climate scenarios? And so Pablo and Dimitris will guide you through that as well. And then coming to the second part, well, what can we do about this? Can we modify the way we do agriculture, and therefore can we evaluate the capacity of different cropping systems, for example, organic cropping systems, to buffer against climate stress. And that we have done basically using an on-field simulation in a long-term field trial. And there, Elena and Fina and Rainer will tell you much more about what we have done in that field trial. So with this, I actually want to conclude here. I stop sharing my screen. I would welcome actually Dimitris on screen, and we would first hear about climate models and predicting change in agroclimatic zones into the future. So please, Dimitris, the floor is yours.
Thank you, Martin. For the introduction, I'm going to start sharing my screen now, and I'm going to give like a small presentation of what we've done on sort of predicting the impact of climate change on agroclimatic zones in Europe. Can you see my screen? Is it like okay, full screen now? My presentation?
Not yet. Yeah, now it is. Excellent.
Okay. So again, thank you for the introduction. I'm Dimitris from the National Observatory of Athens, and for as Martin said, for microservices, one of the tasks we had was to use regional climate models to predict how climate is going to impact European agroclimatic zones. I'm going to start first with an objective. The objective, as stated in the grant agreement, was to map future changes of agroclimatic zones over Europe under two different IPCC representative concentration pathways. So under two IPCC scenarios, RCP 4.5 and RCP 8.5. I'm going to very soon speak about what these scenarios mean in terms of emissions and mitigation measures. I just have to state here that part of this work was published already a couple of years ago by a team that didn't include me. I wasn't part of the team yet, part of the team at the National Observatory of Athens. So when we speak about climatic zones, what do we mean? Climate zones are areas with distinct climates. So that's a very brief but precise sort of definition that allows us, climate zones, classifying the Earth into different distinct climate zones, allows us to understand the conditions in which different regions currently have and track how they might change. You can see this map from produced by the EEA. These are the main climates of Europe. So we classify climates usually in Europe into the subtropical, temperate climates, circumpolar climates, and cold climates. And then we have sort of again another classification: maritime, intermediate, continental, or transitional. These are sort of typical zones, typical climatic zones. Related to climatic zones are agroclimatic zones. These are areas defined by distinct climate conditions that modulate crop health and productivity, and thus, sort of in a socio-ecological system, agricultural practices. Climatic zones are distinct from climate zones, in the sense that they are built using climatic variables that are more related to crop health and productivity. So in our case, what we did, and that's sort of the overall methodology that we used, we extended the work done by Segler in 2019 by implementing additional regional climate model simulations in our analysis. Regional climate models are sort of downscale in simple terms. they involve downscaling global climate models. So a global climate model might have a resolution on the map of 100 kilometers, and a regional climate model, which is sort of centered on a region in the world, like Europe, is called the regional climate model, and it's usually a downscaled version. It's more complex than that, but it's a sort of a downscaled version of a global climate model. In our case, we used so 11 bias-corrected Europe Cortex regional climate model simulations. We took 1981, so this is a mistake, 1981 and to 2010 as a reference period. And we clustered the maps based on two variables, growing season length and active temperature sum, using a k-means clustering algorithm. As I said before, we used two future scenarios, two regional concentration pathways. Regional concentration pathway 4.5, which is the scenario implies moderate mitigation measures to reduce CO2 in the atmosphere, and RCP 8.5, which is sort of an extreme scenario where no mitigation is applied, no mitigation is done to reduce emissions, and emissions keep increasing up to 2100. We did the analysis under two future projection periods, 2031 to 2060 and 2071 to 2100. More specifically, so as I said before, we used 11 regional climate models from Eurocortex, which they have data, there are simulations between 1981 and 2100. They have a 0.11 degree resolution, which in kilometers, it means 11 kilometer gridded maps. These models, the regional climate models, can be thought of as gridded maps or as a time series of gridded maps. So in each grid cell, we calculated an ensemble median for all these regional climate models that you see on the right, for all these Eurocortex regional climate models on the right. We took an ensemble median for two climate variables. And we calculated two climate variables. Growing season length, which is a very important, perhaps the most important climate variable related to agriculture, which is the sum of days between the first time in the year when, for at least 6 consecutive days, the mean daily surface temperature is greater than 5 degrees Celsius, and the first time for at least 6 consecutive days when the mean daily temperature is less than 5 degrees. What this means is it's a sort of a simple algorithm, a threshold-based algorithm that allows us to calculate for how many days in each gridded cell plants can grow. The second variable we calculated, an ensemble median for, was the active temperature sum, which is, again, a very important agroclimatic variable, which is the sum of the daily surface temperatures above 0 degrees during the growing season. And it's the first growing season length is measured in days, while active temperature sum is measured in degrees Celsius. And then, based on this ensemble, and then the method for creating the agroclimatic zones, we split sort of the data set into 3 periods. One of them is the reference period, on which we first extracted median values for growing season length and active temperature sum from the Eurocortex ensemble for all the Eurocortex model. Then, using Lloyd's method, we applied the k-means clustering with a for 8 clusters to recognize, to identify 8 clusters after Segler 2018. And then that way we identified 8 clusters in the maps. And then for the future scenarios, again, we calculated future ensemble median for growing season length and active temperature days, which were then normalized based on the historical reference period that we calculated above. And then for the future scenarios, again, in between 2031 and 2060 and 2071 and 2100, we normalized growing season length and active temperature sum data into one of the clusters that were calculated for the historical reference period based on their distance from them. Moving on to the results. So we created 8 clusters, which have, as you can see, different median growing season length days and active temperature sum values, and we sort of clustered them into 8 zones. We call them after Segler and after many climatic, like using the nomenclature of a typical nomenclature for climatic zone identification and classification. We classified Europe into 8 agroclimatic zones. So starting from the south, Mediterranean, South Maritime, North Maritime, Pannonian, Continental, Nemoral, Boreal South, and Boreal North. In the graph on the right, you can see how these how the 2 agroclimatic variables, growing season length and active temperature sum, change as we move across agroclimatic zones. Like if we read the graph from the left to the right, you can see that growing season length increases going from the Boreal South, from the Boreal North to the Boreal South, to the Nemoral, to the Continental, the Pannonian, to the Mediterranean agroclimatic zone. And the same happens for active temperature sum. So we can see here how the algorithm identified these 8 clusters based on the 2D space created by these 2 agroclimatic variables. So in terms of the map that we are looking for the historic reference period, we classified again into this is how the 8 clusters look in Europe. And as expected, Southern Europe is characterized by the Mediterranean, the South Maritime, and the North Maritime agroclimatic zones. They have a complex spatial distribution, which is related to topography and natural features and barriers, and the Mediterranean Sea, of course, though, but also some mountain range systems that are very prominent in this area, like the Pyrenees or the mountains of Greece or Italy. In Eastern Europe, we can see that the distribution of agroclimatic zones is more homogeneous. Pannonian, Continental, Nemoral, and Boreal South agroclimatic zones follow a distinct gradient, which is largely based on the region's latitude. And in Western Europe, the dominant agroclimatic zones are South Maritime, Northern Maritime, Continental, and Nemoral. And of course, in the north, in the far north of Europe, we have the 2 boreal agroclimatic zones. I mean, the Northern Europe is dominated by these 2 agroclimatic zones. Of course, the results are in agreement with the results of Segler et al. From 2019. What's interesting, of course, for microservices is how these agroclimatic zones are projected to shift. I mean, this is the interesting part, and this is aside from finding itself, it's also an input for other work packages that build on this data to predict how the microbiome is going to change, might change in the future. So for the moderate mitigation scenario, regional pathway 4.5. Gray areas in the map are areas where no change is projected to happen. Okay, so for 2031 to 2060, which is like a few years from now, several European regions are estimated to experience a considerable migration of agroclimatic zones towards the north. Okay, and that's very characteristic in the southern part of Europe, and especially for the 2071, 2100, when this northward signal becomes regionally extended over larger parts of Europe, as you can see in the map, and you see the Mediterranean, for example, climate moving northern and northern. For example, in southern France, but also like northern maritime climates, southern maritime climates moving again close to, I mean, in southern UK or northern France. They're just examples of how climate is predicted to shift under this sort of methodology. And as expected, results for the no mitigation scenario for RCP 8.5 are much more severe. In 2031, 2060, large parts of Europe will experience a considerable migration of agroclimatic zones towards the north. And for RCP 8.5, the results for 2031, 2060 are similar to the model mitigation scenario, but for the longer time scale, for 2051, 2100. Again, in 2071, 2100, a change in agroclimatic zones is projected for almost all parts of Europe, with a more pronounced northward zone migration. Very characteristic, I would say again, is the northward shift of all agroclimatic zones. Like the Pannonian, it moves all the way into Russia. The Mediterranean climate is projected to, if no mitigation measures are taken, is projected to cover almost all of southern Europe, and the southern maritime climate covers almost all of Western Europe, aside from sort of central and southern Germany. So some sort of very small conclusions. A strong northward shift of the agroclimatic zones, especially under the no mitigation scenario, is projected towards the end of the century. A significant shift in the agroclimatic zones is also projected for the moderate mitigation scenario, RCP 4.5, in the near future, and the end of the century period for large areas of Europe. And even the moderate scenario implies shifts in the climatic envelope that is suitable for particular crops, in particular areas of Europe, and it raises concerns about the resilience of microbial communities and microbe-plant interactions. We will hear more about these concerns, more about these changes in the next presentation, I think, by Pablo. Thank you very much for listening.
Thank you, Dimitris, for that insights into the climate model part. So I would like to remind you, please, if you have questions, don't forget there's the Q&A box, so you can enter your questions that we will basically discuss at the very end. And with this, I would like to hand over to Pablo, who will talk about now the soil biodiversity along these agroclimatic zones. Pablo, please, the floor is yours.
Okay. Thank you, Martin, for introduction. Can you see my full screen now?
Yes, full screen, fine.
Okay. Hi, everyone. My name is Pablo Sánchez Cueto, and I will present the predicting soil biodiversity and functionality results across the changing agroclimatic zones that previously Dimitris mentioned. Following up the previous presentation from Martin and also Dimitris, we show that several parts from Europe will experience intense level of warming, leading to continued significant migration of climate zones over the next decades. These changes are particularly worrying in agroecosystems, where this accelerated climate change, in combination with other stressors as can be the unsustainable soil management, directly threatens the food productions in this growing population. So in this line, the soil below ground biodiversity is increasingly seen as a key factor in maintaining the ecosystem multifunctionality, as can be the enhancing crop growth, nutrient use efficiency, or disease resistance. However, the consequences of climate change on soil biodiversity and ecosystem services that they provide, as well as the feedbacks that this biodiversity can have on the climate change, are far from being understood now. So hence, our study aims to address this knowledge gap using the current biodiversity and functionality relationships to anticipate future dynamics in the context of climate change. We summarize here then the two main objectives that we have during this project. The first one is to characterize the shifts in soil biodiversity and functionality along the current climate gradients using the Lucas soil data. And the second is to develop artificial intelligence-based models to forecast region-specific soil biodiversity dynamics and cascading effects on multifunctionality under future climates. Both objectives were reached thanks to intrinsic collaboration with the Lucas Soil Network, which is an European soil initiative with data from several years of sampling campaigns across soils in Europe with different land uses. So here I would like to thank the ERC collaborators, as can be Maeva, Alberto, Panos, and Arwin, due to their high efforts on generate and share this data that is really useful for this type of projects. In terms of the data collection from this dataset, we focus on non-permanent croplands in this 2018 Lucas campaign. For this, we use the prokaryotes and eukaryotes alpha and beta diversity metrics, along with the taxa relative abundance for these 214 sites. In terms of the functionality, this was assessed via enzymatic activity, the plant productivity, and the nutrient availability. Thanks again for the combination efforts between people from this project, the Lucas Soil Network, and also Carlos Guerra team that performed the enzymatic activities. Also, we use climate data from the World BioClim from 1979 to 2018, and this was translated, as previously mentioned, Dimitris, with other models into agroclimatic zones. For future climatic variables projections, we use CMIP climate simulations under different shared socioeconomic scenarios pathways. That these reflect various global development scenarios that is similar to the RSCPs that previously mentioned by Dimitris. In particular, here we focus on the SSP 126 scenario, that is a low emission scenario characterized by strong mitigation efforts, sustainable development, and reduced anthropogenic climate impacts, and the SSP 585, that is a high emission scenario that assumes continued reliance on fossil fuels, leading to more severe impact in climate change and environmental degradation, both across for the year periods. Also, we consider the future agroclimatic zones described by Dimitris in the previous presentation, but this future data is all related with the prediction models that will come at the end of my presentation. And now I will show the results about the links between the current biodiversity and the current climate data. Starting these links, I first present the alpha diversity, which is the number of different taxa found across sites. Here we group the samples by agroclimatic zone, which from left to right, you can see that is the nemoral, continental, Pannonian, North Maritime, South Maritime, and Mediterranean, or which is the same from cold temperate to maritime and Mediterranean places. In this case, if we look at the prokaryotes in the left, we observe high variability within region with no clear gain or loss of biodiversity by any climatic region or zone. But for the fungi, there is also quite variability within the regions, but there are some difference between agroclimatic zones. In terms of, for example, the highest values were found for nemoral and continental soils, contrasted with the lowest for North Maritime and Pannonian sites. which overall this indicate that the fungal diversity is potentially influenced by factors such as temperature, humidity, or the seasonal variation, which these factors vary across the regions, and this can be considered as main drivers of biodiversity. But moving to the community composition, we represent here the beta diversity by a canonical analysis of principal components, where we look at the constrained variance of the biodiversity impacted by these agroclimatic gradient, agroclimatic zones. Here, the influence of the agroclimatic zone is much more marked than in terms of the alpha diversity, where the samples from the let's say the cold temperate climates, as the ones in green from nemoral and continental, are clustered separately compared to the ones from Mediterranean and South Maritime for both types of organisms, but it's much clearer for the prokaryotes. And for example, if we look also at the overall success rate, the prokaryotes present a higher one, which indicates, again, with a numeric term, that the influence is stronger for the prokaryotes community composition than the fungi with this type of analysis. Moreover, if we continue exploring these agroclimatic gradients, we look at the soil communities, let's say, as the soil communities at the taxon specific level. Where in this case, they assess the differential abundance of the OTUs group at the genus level across the different agroclimatic zones. So here we found that around the 50% of the taxa was differentially abundance between the agroclimatic regions for the prokaryotes and the 10% for the fungal genera. And among these, we represent one of the most significant ones and the ones that has relevant mechanisms in the soil. Starting, for example, by Microbiota from Proctobacteria phylum, the bacteria was found to increase in cold temperate climates with a mechanism that they were associated, let's say, with important role in symbiotic nitrogen fixing. So we can see here an association between climate and future, let's say, potential change in nutrient cycle. In contrast, Acidothermus, that is actinobacteria, is increased in Mediterranean maritime soils, which is a thermophilic that degrades plant biomass, and so these are involved in cellulose decomposition. So again, this can be also related with the nutrient cycling. Moving to the fungi, the increase in Exophiala, that is Ascot mycota, in cold temperate climates could indicate an adaptation for this polyextremo tolerant in cold environments. And finally, Fusarium and Alternaria, that are a genera being considered potential pathogens, were found to increase in Mediterranean regions, raising let's say, a potential concern about disease spread under these warm zones. Now moving to functions, mention here now how we generate the multifunctionality index, because sometimes it's not so clear. For this, we first analyze the redundancy between the functions within the soil measurements from the soils to then identify which will go into the multifunctionality index. After assessing this, we decided to include the average standardized values of the nutrient availability, which is the organic carbon, nitrogen, and potassium in this measuring the soil, the enzymatic activity, which is the siloxidase, cellulase, beta-glucosidase, and acid phosphatase. And in the terms of plant productivity, we use only this NDVI index that is from Earth observation measurements. And at this point, what we call multifunctionality and what we will use to characterize and then predict based on the bacterial abundance. So when look at the soil functionality patterns across the climates, there is no clear gradient as we observe in the community composition for the biodiversity, with high variation in terms of function within regions. It's interesting to say that soils from Nemoral, for example, but also from South Maritime, presenting higher multifunctionality than Mediterranean, as an example that there is not a gradient for this case. Then, up to this point, interesting agroclimatic patterns emerge for several aspects in soil biodiversity, which are clear in terms of composition than for the alpha diversity and for the functionality in the soils. So, in particular, this composition is quite remarkable for the differential bacterial genera abundance. So the next part of my presentation, I will focus on how we assess the predictions of this soil bacteria abundance based on climate data. To achieve that, we implement what we call artificial intelligence methods. And let's say divide it in two steps and with a driven cascade. We call a driven cascade because the first model predictions are influencing the second model ones. And here we perform this to forecast what would happen in a changing climate. But focus on the first step about the prediction of bacterial relative abundance at the genome level that we generate based only on climate data. Importantly, for this machine learning, we use the 70% of the dataset for training and the rest 30% for test and validation. And also, as you can see here, that several machine learning methods has been tested, as can be the random forest, decision trees, or lasso regression. All the final results are based on random forest because this approach gave us the best performance in terms of model predictions accuracy. To discuss a bit the models, I will show you only the long-term predictions for the range of 2081 to 2100. That it's important to note here that we use one model for climate scenario to predict the bacterial abundance per each one. And navigating a bit the slide, in the left, you can observe a summary of the model error by scenario, with the majority of bacterial abundance predictions maintaining a high accuracy, with most cases falling below 1% of relative error threshold, which inform us about the robustness of the created models. And this is particularly evident under the optimistic scenario. However, if we look at the pessimistic scenario, the one with SSP 585, we can observe a larger proportion of bacteria with high relative error, which means a high uncertainty that can be induced by this high emission scenario, as could be expected by more drastic climate shifts due to no mitigation actions, left to more difficult predicting changes in terms of bacterial community composition. And on the right, to further illustrate the performance of the predictive models, we present some examples of the graphical comparison between predicted and actual bacterial abundance values. In this case, we represent some examples from previous mentioned bacteria as Acheothermus and Microbiota. In this type of graphic, the red line is the current abundance and the yellow line is the predicted, with the distance between them reflecting the standard error, with at the end show the prediction reliability across these models. What we can see in this example is that the yellow line matches quite well with the red line, making these predictions, along with the other or most of the other genera, high accurate. At this point, we can see that the models from all bacteria was validated and tested. So we can move to the second step where the matrix of predict bacteria genera were used to predict the future change in terms of multifunctionality, or also called the cascading effects on this multifunctionality. This second step was performed based on a model linking the current bacterial abundance with the current multifunctionality by site, to then forecast the cascading effect that future shift in this bacteria abundance will have on multifunctionality. Again, the final model was developed based on random forest due to the best performance across them. The model developed in this second step allow us to forecast how predicting shifts on microbial taxa could have cascading effect into change in ecosystem functions and at each site for the two socioeconomic scenarios that we mentioned at the beginning. In this case, to simplify the interpretation, we group in the figure that you observe the multifunctionality predictions values by sites expected to experience significant climate shifts, sorry. At this point, linking this analysis to the predictions that the Noah's and Dimitri's team did at the beginning. And we focus, and here we present, for example, transitions from sites from nemoral to continental, from group 2 to 3, or even from, let's say, continental to South Maritime at group 3 to 6. And what we can observe is that across all climate transitions, the multifunctionality, in this case understood as the mean values of nutrient availability, enzymatic activity, and plant productivity, is predicted to be higher under the optimistic scenario, the green one, the green bars, and this scenario that is also called good or green scenario, compared to the high scenario in yellow that is called bad, or also the fuel-dependent scenario. Which means that this trend suggests the low emission scenario could better preserve soil multifunctionality across Europe. Moreover, we did analysis to observe which of the bacteria can be mediating with highest importance this multifunctionality, and we identify some of the genera as Nitrosphaera, Niabella, Cribella, Agromyces, Rombustia, and Terrimonas that at this point can suggest to further investigation of what could be the role of these organisms and their future climate change and how this can impact the functionality. So then, to sum up our study, where we combine the molecular biodiversity, climate data, soil functions, and machine learning methods, we can conclude that the agroclimatic gradients influence soil biodiversity composition, particularly among the bacterial taxa. The artificial intelligence models can accurately predict change in the abundance of a specific bacterial taxa based on current and projected climate data. And the cascading effects of bacterial taxa shifts on soil multifunctionality were distinct depending on the shared socioeconomic scenarios under study. The green socioeconomic pathways, the models associated with this pathway are consistently predicting higher soil multifunctionality across Europe, compared to the fossil fuel ones. And at this point, I would like to finish my presentation, and we're happy to answer any questions at the end. I will.
Thank you very much, Pablo, for these interesting insights. And with this, without much, I would just pass it on again to Dimitris, who will show how they have also implemented more of the Sentinel-2 Earth observation data into this prediction. So please, Dimitris, take it on.
Yes, thank you, Martin, again. Please let me know if you can see my screen, my full screen.
Yes, not yet the full one, but yes, now it's the full. Perfect.
Okay. Yeah. So this presentation is about developing a pipeline that builds on the same data set, the same Lucas data set that Pablo discussed, and to explore whether we can use Earth observation or Earth observation and meteorological data to predict aspects of bacteria diversity in European permanent croplands. So in this presentation, building on work done for microservices that explore correlations between Earth observation indices and aspects of biodiversity. Here, in this presentation, which is subsequent work, we explore whether soil bacterial diversity indices can be accurately modeled using a combination of Earth observation and meteorological data set without relying on ground measured soil or anthropogenic variables. So in this sense, we use the Lucas data set for bacteria diversity, and Earth observation and meteorological data. So again, we use the Lucas 2018 survey that Pablo talked about, 2014 non-permanent cropland samples, and we use different indices of alpha and beta diversity. We map them across Europe, and this is just, you can see two examples here where we have mapped, observed the ODUs and the first axis of a principal coordinate analysis for the Lucas 18 samples across Europe. Again, there are 8 indices. For alpha diversity, it's observed ODUs, COW1, ACE, and Shannon. And for beta diversity, it's the first 2 axes of a principal coordinates analysis and the first 2 axes of a canonical analysis of principal coordinates axis. We use 12 Earth observation indices. We take a 3-month mean around the sample date of the Lucas sample date. We derive them from a Sentinel-2 optical satellite, and these indices are related to productivity and health, moisture and soil composition. I'm not going to go through them now, but they're related to these 3 sort of characteristics of the landscape. And then we also built models using both Earth observation and climatic variables. For climatic variables, we used precipitation, soil moisture, drought severity, solar radiation, minimum temperature, maximum temperature, average, and covariative variation. And we built 3 types of models: random forest, cubist, and elastic net regression, and for all the soil biodiversity indices I mentioned before. So for the experiment I'm presenting sort of today, again, it's part of a larger experiment, but for what I'm presenting today, it's 2 types of models: Earth observation only, or Earth observation and climate. Okay, so what do we find? We found that Earth observation only models are able to predict aspects of bacterial diversity, beta bacterial diversity mostly. So if you see the R squared on these tables on the left, you can see that for alpha diversity, it's not very good, up to 10% of the variation for the Shannon index. While for beta diversity, using again Earth observation only models, and that's only optical satellites. We could use other satellites that would give us more information regarding temperature and moisture, etc. But that's sort of for the next steps I'm going to discuss before. But using Earth observation only models for beta diversity, we're able to have R-squared values of close to 0.4 or 0.26 for the axis of the principal coordinates analysis and the canonical analysis of principal components for the Lucas dataset. Now, using both Earth observation and meteorological variables, the models were significantly better, I would say, up to close to 0.9, 0.87 R-squared values for the first axis of the canonical analysis of principal coordinates. While for alpha diversity, again, the values were better, up to and not bad, like up to 20%, close to 10% of the variance for the COW1 index, which is not bad. So we conclude here that, especially as we take this methodology further and we use larger timescales, more data, more different sensors for the satellites that are able to capture more climatic variables like land surface temperature or soil moisture, etc., we could start building a pipeline that's able to accurately predict aspects of bacterial diversity from space. And this, you can see a better a representation of how good the models were for Earth observation only models and Earth observation and climate models. For alpha diversity on the left, you can see that Earth observation and climate models were better, but only slightly for alpha diversity. While for beta diversity, Earth observation and climate models for all biodiversity indices were much better based on the for the error in my error. Now, this is an interesting result regarding variable importance, which we also calculated based on the models. And especially if we look between alpha and beta diversity, I'm going to start by saying that the best Earth observation index is an index. It's called NDCI, and it's related to calcareous soils, and it seems to be to calcium in soils, to natric soils, and this is interesting that this index appears to be the best index for almost all biodiversity indices that we looked at. It appears in almost all variable importance plots. A second observation is that almost all Earth observation indices that were included in the top 20 importance features were almost all related to soil characteristics and not vegetation characteristics. That's another interesting finding here. And and then maybe a sort of microecological finding relates to which climatic parameters were more important for alpha and beta diversity. For alpha diversity for observed ACE and K1, you can see that soil coefficient of variation seems to be the most important variable shaping alpha diversity, while for beta diversity indices, it seems to be temperature aside from it seems to be temperature and precipitation, but mainly temperature. So, in conclusion, I think we would say that Earth observation has the potential to advance operational monitoring of soil bacterial diversity at a continental, at least, scale. Beta diversity models, in our case, were much better than alpha diversity models, with some very promising results by combining Earth observation and climatic data. For Earth observation indices, it was mainly indices related to soil characteristics that seem to be most relevant. This index, the normalized difference index for calcareous and sedimentary rocks, seems to be the most relevant index across related to bacterial diversity. And of course, climatic parameters seem to capture patterns of beta, but not alpha diversity. If you recall, alpha diversity models were not very, the accuracy, the results, R-squared of alpha diversity models was not that high, and that's reflected in the results, if you remember, by Pablo, who didn't find sort of large differences in alpha diversity between climatic zones. Again, this is part of a larger experiment. I showed you just part of it. The results that we have submitted for a publication in a journal related to remote sensing. The results are part of a larger experiment. We explored different algorithms, different climate data windows, different Earth observation data windows. We explore the possibility of using just Earth observation indices that come from bare soil composites when the soil is bare. Like in total, more than 2,000 different models we're exploring this in this manuscript, which is submitted for publication. And regarding future work, as my time has run out, we are trying to build pure Earth observation pipelines by combining sort of sentinel optical satellites with radar, for example, satellites that are able to capture soil moisture better, or Landsat satellites that can capture land surface temperature better. We are putting an emphasis on time series, so using Earth observation data as a time series in the models by using specific algorithms or models that are able to take time series data as input to predict something extrinsic, like functional scalar models, extrinsic time series regression, and other sort of types of algorithms and models. We are also currently building bigger data sets, combining different data sets on microbial diversity, compiling them, aggregating them, and that allows us actually to run more complex algorithms on this data. So deep learning architectures, computer vision-based architectures. And I think we will see some promising results regarding operational microbial diversity monitoring from space pretty soon. Thank you.
Thanks a lot, Dimitris, and also Pablo for basically showcasing how climate will influence basically soil biodiversity and functionality, and how we can use climate models and Earth observation data together with existing biodiversity data to forecast a bit what will happen in the future. So if you have questions to that topic, don't forget there's the Q&A box. Please put in your questions. With this, we have finished the first part. I would like to go on to the second part, and now the question is really if climate change and climate extremes are changing soil biodiversity and functionality. Can we basically use agriculture management practices or the entire management systems to basically modulate these responses? And with this, we will actually go to 3 talks about a long-term field trial that will basically now be introduced by Elena along with what she has done. So please, Elena, the floor is yours.
Thank you, Martin. I will share my presentation. I hope you see full screen.
Yes, we see it.
Good. So welcome from my side. I will present today the first part of the field experiment. So this field experiment was running in the DOC long-term trial, and this trial is running since 1978, so almost since 50 years, and is currently in the 7th crop rotation. It compares 5 different systems, so one unfertilized control, which received no input since almost the beginning. Then there are 2 organic systems, a biodynamic and a bioorganic. They have a crop protection done mechanically, indirect, and with beneficials. And while the biodynamic receives composted manure as fertilization, the bioorganic receives rotten manure. Opposed to that, there are 2 conventional systems, one being fertilized with a combination of stacked manure and mineral fertilizer, and the other one exclusively minerally fertilized. Those 2 have crop protection done with insecticides, fungicides, and herbicides. And previous studies have shown that these systems differ quite heavily in their soil parameters. So, for example, here with some soil biological parameters. First, we see the microbial biomass being the highest in the organic system, especially in the bioorganic system, followed from the mixed conventional system and the mineral conventional system. Next, we see the microbial diversity, not strong difference, but still significant difference, following the same pattern with the organic than the conventional systems. Lastly, we also see that activity measured here as respiration or phosphatase, as well as the heterogeneous activity, we see again the highest activity in the biodynamic system, followed from bioorganic and the mixed conventional system, and last, the lowest in the mineral conventional system. But not only the biological parameters differ, but also the soil chemical. For example, here with the soil carbon stocks. We see here on the x-axis over time. And here we see almost in the beginning of the trial, they were quite similar. And then with the time, they actually diverge. And we see here the carbon stock change. So while the stockless system lose carbon over time, the mixed conventional has a balanced carbon stock, and the organic system gain carbon over time, especially the biodynamic system. And it has been proposed that higher soil organic carbon concentration can result in a higher soil water retention. On the other hand, we also saw, or studies showed that these systems have distinct soil microbiomes. Here on the right side, assessed with DNA-based methods, the taxonomic composition, we see distinct composition in each cropping system. And on the other hand, a quite recent study showed that this is also the case for the soil's functional potential. As we see here, all the cropping systems have also a distinct soil functional potential. And therefore, we then ask ourselves if these different communities or soil function potential have different abilities to resist or resile towards drought. So for this, then we did a drought simulation experiment using these rainout shelters. They were installed in winter wheat. The rainout shelter experiment was conducted in 2022. On one side, you see the rain-fed control. On the other side, we have the drought-induced plots, and we installed them in 3 very contrasting systems in the biodynamic, the mixed conventional, and the mineral conventional system. So going a bit to the results, we see here the soil gravimetric water content for the different sampling time points. So we sampled at stem elongation, flowering, ripening, and 2 resilience samplings. Here before the resilience samplings, the wheat was harvested, then we removed the rainout shelters and we did a re-wetting. For these 3 time points in the beginning, we could sample roots, so including rhizoplane and endosphere, and rhizosphere. For the resilience, we only have the bulk soil samples. So during the drought period, we see here that we have a significant lower gravimetric soil water content during this drought period. Then after the re-wetting, we see an increase, however, still a significant difference, which disappears after 11 weeks. If we look at here, we see that we have around 10% water content in the soil. So comparing that to literature, we could see that we could induce actually a severe drought. So now we wanted to see, on one hand, which species were affected by drought. And for this, we used a metabarcoding approach using the 16S RNA and ITS amplicon. And then for the metabolic potential, we used a shotgun metagenomics approach. Since the rhizodeposition is quite an important link between the microbes and the plant, we wanted to also have a glimpse what is happening with the rhizodeposition. And for this, we did a semi-continuous stable isotope labeling with 13 CO2s to see a bit about the carbon going into the soil from the plant. So here you see the taxonomic results for the prokaryotes and the fungi. Here in the points for the drought, and in the stars for the rain-fed control for all the cropping system. And we can nicely see, and this is rhizosphere data. We can nicely see that these cropping systems have, again, distinct soil microbiomes, but also we see that we have a drought effect on the soil microbial communities. And interestingly, the extent of the shift is quite similar between all these cropping systems. As a next result, I would like to show you the Permanova results. So the R squared, the variation explained by a certain effect. So here the drought effect for the prokaryotes and for the fungi. We see here also the significance level, and we can see for the bulk soil and the rhizosphere that the drought effect was actually stronger in this agricultural site for the fungi compared to the prokaryotes. Also, what we can see is that the drought effect actually increases from the bulk soil towards the root, so towards a closer plant association, especially for the prokaryotes, but also slightly for the fungi. What is interesting is that even though the extent of the shift is quite similar between the cropping system, we still see a unique microbial composition even under the severe drought stress, and that is showing that there is still a strong management legacy even under severe stress. As a next step, we wanted to see a bit about the cropping system specific responses of some genera, and here you see the whole graph, but that's quite a big graph and everything is a bit small. So I would like to highlight some responses of plant growth promoting microbes. First, I would like to show Rhizophagus, which was relatively more abundant under drought, especially in the biodynamic system. This is an arbuscular mycorrhiza fungi involved in the nutrient and water uptake and improving that for plants, also being able to reduce oxidative stress and being able to regulate the aquaporine gene expression. As a next, there is Vavoriax, a protobacteria, which was especially relatively increased under drought in the mixed conventional system. This bacteria is known to produce auxin, hydrophores, as well as the ACC deaminase. It's also able to reduce the abscisic acid as well as the oxidative stress in plants. Lastly, one very well-known plant growth promoting bacteria, Streptomyces, was especially more abundant under drought in the conventional system, especially in the mineral conventional system. This Actinobacteriota can produce auxin, hydrophores, and the ACC deaminase, as well as it's able to fix nitrogen. So having this in mind, we then wanted to go to the soil function potential, and we assumed that this might be still distinct, potentially exhibit different abilities or different potentials to resist against drought. So we indeed found that the soil function potential has actually quite a similar picture as the microbial community composition. We still have this unique systems, cropping system specific response, but we also see that there is a drought effect on this soil function potential. So we wanted to, on one hand, see the general adaptation mechanism having this soil function potential of the microbes, but also on the other hand to see if there is an indicator gene of the drought response of one cropping system. However, the second part, the indicator gene, is not yet ready to present. So for this, you have to stay updated to see the results hopefully soon. So for the general response across the cropping system, we saw that we have an increased potential for osmolyte accumulation, for example, for the monosyl glucosyl glycerate, as well as ectoin and betaine and proline. osmotic accumulation is a well-known adaptive mechanism against osmotic stress for microbes, for soil microbes. On the other hand, it's also known that microbes can form spores as well, become dormant in response to drought stress. So indeed, we found that genes associated with sporulation were relatively more abundant under drought, underlining that adaptive mechanism. However, we found that, for example, genes related to ribosomal hibernation were relatively decreased under drought. And ribosomal hibernation is actually a mechanism to focus only on the essential protein synthesis under drought, since that is quite costly for them. So you would expect that this is actually increased under drought or under stress. So this decrease, it's not known, but we currently assume that might be rhizosphere specific, since there are many plant growth promoting microbes in the rhizosphere helping the plants to endure the drought. So the plant has maybe a potential interest to keep them active and profit from their phytohormone or hormone or cytophore production. On the other hand, microbes can produce exopolic saccharides to endure osmotic stress. However, we also found that cholinic acid, which is an exopolysaccharide, was relatively decreased under droughts. So that might be also rhizosphere specific, since in the rhizosphere there is already mucilage produced by the plant. And so for this, they can reduce the cost of their exopolysaccharide synthesis and profit from the mucilage excreted by plants. However, that is all not known. It's just currently our assumption why that is so contradictory to other results. So for this, then we wanted to make the link to the plant. So is there something happening also with the plant that could indicate that there is some rhizosphere specific response? And indeed, we found an increase in rhizodeposition carbon. So here we see the carbon in the absolute value, and here the relative. We see it for the grain, straw, root, and the rhizodeposition for the 3 different systems for rain-fed control and drought. And we can indeed see that there is an allocation of carbon to below ground, and especially we see that the rhizodeposition increases in all the systems, potentially influencing the rhizosphere microbes and interacting maybe with the plant growth promoting microbes. On the other hand, we also looked at the root carbon biomass and looking at the coarse roots as well as the fine roots in the system, in the drought and in the rain-fed control. And we found that there is an increase in root carbon biomass, especially in the fine root carbon mass, and specifically in the farmyard fertilized system. We currently don't know why that's the case. Recent or previous studies have shown that these systems have increased organic matter, as we saw in the beginning, and that could translate in improved soil structure, porosity, nutrient availability, potentially leading then to an improved root growth under drought as well, so helping the plants to adapt to these low nutrient as well as water limited conditions. So then to the take-home messages, we see that drought affects soil microbial communities in this agricultural trial. It increases with closer plant associations, so from bulk soil rhizosphere to the root, and we found that fungi are more sensitive towards drought in the bulk soil as well as the rhizosphere. Secondly, we couldn't find any indication that the increased soil organic carbon in the organic system could enhance the resistance and resilience of soil biodiversity towards drought. So this shift, the extent of the shift, was not different between the systems. However, we found that we find still distinct soil micro communities in this cropping system under severe drought stress, underlining that there is an important legacy effect of this agriculture management on the soil biodiversity persisting in stress conditions. And lastly, the riser deposition carbon increased under drought, and that has potential implications for carbon storage and rhizosphere microbes, possibly interacting with these plant growth promoting microbes, but there are still further studies to do to see if that's the case. And with this, I would like to thank all the people involved in the trial and the funding of Biodiversa and SNF, and you for your attention. Thank you.
Thank you very much, Elena, for these interesting insights. Please, if you have questions for Elena, put them in the Q&A box. And I would like to proceed with Fina, and Fina has looked at a very specific component of this biodiversity, namely at the nitrogen cycling microbial gills. So please, Fina, if you could turn on your camera, share your screen.
Okay. Martin, I think you have to start my video because. Okay.
Excellent.
Okay, is it okay?
That looks good.
Okay, thank you everyone. Good morning, and I'm Fina Bintarti. I was working at INRAE with Laurent Filippo on the work package 4, and specifically we will look at the nitrogen cycling microbial communities under drought stress. And we found that they respond to drought actually modulated by the cropping system. Just a little bit of background that the climate change is happening, and the future climate projection showed an increasing drought frequency and intensity by the end of 21st century. And severe drought has been reported across Europe in 2018 and more recently in 2022. And such an extreme drought can alter the microbial community structure in soil, including those that are responsible for the nitrogen cycling. And droughts can also directly affect the nitrogen cycle, which altogether might have consequences to ecosystem function. As we know that the nitrogen cycle is a fundamental component of a global biogeochemical cycles, and which eventually will affect the crop productivity and quality. So it is important to understand the response of the dysfunctional group of the nitrogen cycling community to drought, to better understand or to better predict the impact of climate change on the ecosystem function and service. And for this specific study, we focus on the nitrification, which is the ammonium oxidation to nitrite and then to nitrate, because nitrification contribute to N loss to the environment through the N2O emission and also nitrate leaching. And also this process is aerobic, so it's very sensitive to changes in soil moisture. And more focus on the ammonia oxidation step, which is the first step of the nitrification, which mediated by 3 different group of microbes, ammonia oxidizing bacteria, archaea, and 1 group of bacteria that capable to do a complete ammonia oxidation from ammonium to nitrates, or we call it COMAMOC. And study has been reported that this group actually very sensitive to environmental changes, so it's a very good and relevant bioindicators for monitoring soil health and also soil status. So for the specific objective, again, for this study is to investigate the response of the ammonia oxidizers to drought, and also to understand to what extent that the different cropping system influence their response to drought. And this study is actually part of ELENA study in the rainout shelter experiment in dog field trial. So the soil were collected in the same time points, and at INRAE we performed the ammonia oxidizer assessment, including the diversity and composition by sequencing the MOA gene by Illumina MiCIC, and then we also analyzed the gene abundance using the qPCR. And we also analyzed the mineral end pulse and N2O flux. And for the result, we saw that a drought strongly affect the mineral end pulse, as shown by the box plot here, the panel A and B. We can see that drought increased the content of ammonium and nitrates, especially in the confirm and conmin. And after the rewetting process, actually we can see that the effect of stress were present first week after the rewetting process, but then the effect of drought was lost after 11 weeks after rewetting. This indicating that the actually the resilience of the microbial and process in that system. And in the biodyn or the organic system, actually the content of ammonium and nitrates were mainly unaffected. And compared to the mineral end pulse, the effect of drought on the N2O flux on the panel C here actually weaker. In particular, we can see that the effect of drought on the N2O flux, drought decrease the N2O flux in the beginning of drought period, also in the confirm and conmin, but not in the biodyn system. When we look at the structure of the ammonia oxidizing communities of the three groups of microorganism, we see that, so this is the ordination plot. So basically points that cluster together are more similar to each other. And the important thing we can see here that the actually cropping system strongly influence the structure of the ammonia oxidizing communities, as we see clustering by color. And then if we look at the clustering within each cropping system by shape is the control and drought treatment. We see that LA, for example, has clear separation between control and droughts. And also AOA has the highest reclassification rate compared to the other groups, meaning that this group is the most sensitive to drought. And we also can say that the effect of drought on the ammonia oxidizing community structure is group specific and differ by cropping system. Next, we want to identify the ASV that change in abundance between control and drought within each cropping system. So the panel A here is the heat map of those drought affected ASV, and the B panel is the percentage of those drought affected as ASV. And we can see that around 20 to 40% of those ASV are affected by drought. And even though on the heat map, we couldn't see consistent ASV that sieve in response to drought across time point, we identify that those drought affected ASV actually belongs to a dominant taxa, which suggesting that actually the dominant taxa not necessarily resistant to drought. And then we also perform a analysis of a abundance of MOA gene copy numbers as a proxy of the ammonia oxidizing community abundance. Actually, we don't we only see a marginal effect of drought, especially in the Komamo clade B, where the drought reduce the abundance of Komamo clade B consistently in the three cropping system, especially in the confirm cropping system. And in this, we also can say that the drought effect also differ among cropping system. To better understand the relationship between the ammonia oxidizing communities and pools and N2O flux and also soil properties, we perform a correlation analysis between those variables. And the important point here is that in the control treatment, we can see very strong relationship or correlation between several ammonia oxidizing community variables with the mineral and pools and N2O flux. But then those magnitude of correlation were reduced in the drought treatment, suggesting that the drought actually reduce the overall metabolic activity of the microbes, including the nitrification. So to sum up of our study, we found that the nitrogen pools are more resistant to drought in the organic or the biodynamic cropping system. And the effect of drought on ammonia oxidizing community is driven by the cropping system. The community structure of AOA and Komamok are more affected by drought than AOB, indicating that the effect of drought is group specific, and droughts also alter or change the relationship between ammonia oxidizers, mineral and pools, and N2O flux. So for this, I want to thank everyone involved in this project, and thank you for your attention.
Thank you, Fina, for these insights into the nitrogen cycling guilds. And with this, we're already moving on to the last speaker, which is Rainer Jürgensen, who will talk about riser deposition and primarily also exopolymeric substances. So please, Rainer, the floor is yours.
Yeah, can you see the screen? Is it fine?
Not yet in presentation mode.
Yeah, now?
Yes.
Yeah? Okay.
Good to go.
Yeah. Thank you very much, Martin, for introduction. So I think the co-authors, Tabata, Rebecca, Anke, I think it's the core group of Witzenhausen. We, of course, had close cooperation with Agroscope and particularly also the FIBE. And I think the background is already presented by the previous presentations, in particular Elena. And so riser deposits feeds the rhizosphere microbiome, and the riser deposits are transferred to the soil microbial biomass and soil organic matter. And this soil organic matter was fractionated by the current project into extracellular polymeric substances derived from plant mucilage and microbial biofilm-like components and particulate organic matter derived from fine root residues. And we also tried amino sugars as indicators of microbial necromass. So we measured experimental draft has been shown, and wheat plants were labeled additionally with 15N leaf feeding, and we measured microbial biomass and biofumigation extraction, EPS with cation exchange resin extraction according to Redmile-Gordon. And in these EPS extracts, we measured EPS protein and EPS carbohydrates. Particulate organic matter was measured by density and size fractionation according to Magid and Kjaergaard, and amino sugars have been measured by according to Indorf with HPLC, and Zhang and Amelung for C13 specific amino sugars according to Zhang and Amelung. So I will 3 parts: sampling plot results from the whole plot, microplot, bulk results, I think, which have been shown by Elena, and microplot rhizosphere results finally. So the sampling plots have been already published this year in Biology, Fertility and Soils, and I will summarize it shortly. EPS contents, you see, are rather constant during the season. A drought leads to higher EPS carbohydrate contents at stem elongation, and drought leads to higher EPS protein contents at ripening. And I think the effects of the different land use systems are, I think, small. And so in the future, drought effects are already there at stem elongation, and in the future, drought effects should be investigated preferentially before stem elongation, as soon as possible after germination, as soon as it's possible to measure, I think, rhizosphere, or to get rhizosphere samples. Yeah, in contrast to the EPS, the ratio of EPS to microbial biomass showed significant system effects. I think Elena showed, I think, the information of the microbial biomass, which is typical, and then the ratios of EPS carbohydrates to MBC and EPS protein to MBN were highest in the ConMin and lowest in the biodin system. So this means the higher the SOC, probably the higher SOC content of the biodin land use system requires less formation of EPS, and in this case, more substrate derived from the rhizodeposits could be diverted into microbial biomass C and microbial biomass N in the biodin system, partly explaining the higher MBC level in the biodin system. So the second part, I think it's from the microplots, I think, where we use the C13 and 15N labeling. And I think on the first two figures on the left side, you see microbial biomass C and particulate organic carbon down the soil profile. You see MBC and POM-C decline with depth and increase under drought. And I think in brackets MBN and POM-N, I think, behaved in a similar way. And MBC and MBN were highest in the biodin and lowest in the ConMin system, not shown. I think it's similar to the information you already received from Elena. I think on this slide, you see the POM-C13 and POM-N15 derived from rhizodeposition. And I think for both, it's for POM-C13 and POM-15, and the contribution of rhizodeposition slightly increases with depth, particularly in the control, and huge variation between the replicates for both fractions, particularly at the lowest depth, at 50 to 75 centimeter depth. Yeah, the contribution of rhizodeposition generally declines with depth and was larger under drought, particularly at 25 to 75 centimeter. And I think there is a significant higher contribution of the rhizodeposits to the microbial biomass, but you should be aware that the microbial biomass at these two depths is much lower than in the surface. But generally, drought increases the contribution of rhizodeposits to the soil microbial biomass. Yeah, the EPS carbohydrates slightly increase with depth. And on the left side, carbohydrates have a lower concentration than EPS protein. And the EPS protein slightly increase with depth, with a strong tendency to be higher under drought. So this is somewhat surprising, because I think the microbial biomass at lower depth is much lower. So, I think this needs some further evaluation. And but apparently the turnover of the EPS is slower, as shown by the EPS C13 and EPS 15N derived from rhizodeposition. I think the contribution of rhizodeposition declines with depth in a small range on a high level for EPS C13, And I think this decline was stronger for EPS 15N, with a strong then tendency to be high under drought, particularly at the lowest layer. So the low layer respond also quite strongly to drought effects. When we look at the atom percent excess for C13, 15N in the two fractions, microbiomes, EPS, particulate organic matter, the rhizotopic derived C13 increases in the order microbiomes, particulate organic matter, and EPS, the rhizotopic derived 15N has a quite different order. It is in, I think, the lowest concentration of rhizotopes are found in the EPS and then microbiomes, and the largest in the particulate organic matter derived from the roots, largely derived from the roots. Yeah, and now I think my last slide show the EPS carbohydrates, the EPS protein, the ratio of EPS carbohydrates to soil organic carbon, EPS C13, EPS 15, and comparing rhizosphere and bulk soil. And the rhizosphere soil contains 9 times more EPS carbohydrates, 4 times more EPS protein, and the EPS carbohydrates SOC ratio was 7 times higher. In contrast, the contribution of C13 was roughly identical, and that of 15N was 86% lower than the respective fractions of the bulk soil. So I think this shows that the rhizosphere is really the hotspot of substrate availability, of microbial growth and activity, and I think indicating that a strong focus should be on this part of the soil. Yeah, thank you for listening.
Thank you very much, Rainer, for these interesting insights into the rhizodeposition effects on EPS and biomass. So with this, we are at the end of all the presentations. I would ask Rainer to stop sharing the screen and all the panelists to actually turn on their camera. I would like now to use the 15 minutes for the different questions that popped up, and I see that actually a lot of the questions came up for the first part, for this the climate prediction and effects on biodiversity. So maybe we can start with a first question, maybe actually to probably best Pablo first. So that you have basically shown these shifts in soil biodiversity, but not really in the functions that you have measured along these agroclimatic zones. And now the question was there more, do we measure the right thing, right? I mean, that participant mentioned that this has also been seen in other studies. So do we measure the wrong thing for the functions? Should we start looking at the metabolic rates of these functions rather than, you know, basically just the values? What is your opinion about this?
Yeah, about the metabolic rate is depend on what we mean with that. Maybe with metabolomics or metatranscriptomic will be a nice approach to improve our understanding with this. Indeed, as in the human being health studies that they want to combine all the multiomics, maybe we have to move to this to really understand what is happening in the soil. This will be complex and is even more if we want to have these global data sets that are aiming to understand this. But that can be a solution in terms of the difference patterns between the functions and biodiversity. This is true, but this is true in terms of the global patterns. And what we demonstrate with applying machine learning methods with deep learning and more, let's say, not sophisticated, but more elaborated analytical frameworks, you can find out more relationships than the first one between biodiversity, between some of the genera and the functions itself. So at the end, this can give you also some information about the links between biodiversity and functionality in terms of predicting with some models. So I think we need also to explore other things in the soil science, to explore other omics. But the combination with these machine learning methods also point out like a good, let's say, perspective for these links that are not yet very understood in the soils.
So what would then be your answer to actually the second question, that how useful is actually soil biodiversity as a feature to predict changes in ecosystem services? Do we need it even, or do we just basically should look at functions?
Yeah, I mean, the biodiversity, if we can give a intrinsic value to itself, that biodiversity as the different organisms that inhabit in the soils, this is a valid point. But then to link with the functions, yeah, there are some constraining, but there are some studies that are able to link this biodiversity to functions. So there is potential to keep understanding better what is the key features of the biodiversity. Maybe the alpha diversity is not the case. Maybe we have to move to these compositional metrics that are more difficult to handle, but can give us more information about the functional shifts in the soils. So that will be, the biodiversity is useful, but maybe not focus only in the richness metrics of the different organisms that we target, but combining different metrics related with this biodiversity in the soil.
Okay, thank you, Pablo.
Thank you.
Then maybe one question toward Dimitris. So there's the question that, have you considered whether integrating crop type management practices such as cover cropping, fertilization, tillage, and so forth could strengthen the relationship between these earth observation derived indicators and soil biodiversity? Yeah, how we could combine these to make even stronger predictions maybe, and is this even, I guess from my side, is this even feasible that we could integrate this at a large scale?
I agree. I mean, the models would be better if we included sort of crop type or management practices into the models. Like I would raise, firstly, I would sort of say that this wasn't our interest. So we're trying to develop, as I sort of mentioned in the presentation, we're trying to develop Earth observation only pipelines, mainly because these are global scale time series data. So they can capture both scale in terms of the whole world, but also in terms of changes. So crop types, you cannot have easily crop type data or soil type data or management type data at the global scale at a very good resolution, and so in a timely manner every 6 months or every 3 months or certainly not at the resolution of a Sentinel satellite every 6 days if there's clouds, or every 3 days if there are no clouds. I mean, there's these two scales that are useful when we are dealing with sort of big Earth observation data that satellites can offer. And that's why we're moving towards this approach as a research team. I mean, the other approach is also valid, especially if you're looking for sort of causal relationships. If you're looking to find sort of how do management practices influence the aspects of soil microbial diversity, then probably the approach suggested by the attendee, I don't remember the name now of the attendee, but the approach suggested would be much more sort of sensible to take that approach. But in terms of monitoring, in terms of timely, regular, global monitoring, I think Earth observation data has this sort of advantage of scale and frequency.
Thank you, Dimitris. And maybe to be clear here, the Lucas soil data, they don't have information on management practices in there. So that's a downside. Otherwise, we probably would have tried how much that would increase our prediction accuracy, right? So, but it's a very good question. I think we can reply. Yes. Dimitris, do you want to?
No, but final. So we did try, like, and others have tried to use sort of soil data from the Lucas, like soil parameter data from the Lucas data set, like pH or electrical conductivity, soil type, and they do produce, sometimes they do produce better models. But although we had this data, I mean, it was a conscious decision because you cannot find this data. You can find them in the Lucas data set, but you cannot find them anywhere else, almost nowhere else in the world at this sort of scale. Thank you for the question.
Yeah, thank you. And then maybe related to this, and well, that question was also for the first part, it actually goes to everyone, because we have looked a bit at also depth effects, right? But the question is, okay, moisture and temperature are varying with soil depth. So do we need to include these in these climate models for predicting changes in biodiversity with soil depth, and how important is soil depth to structure all these properties? So maybe I give that first to Pablo and Dimitris, and then I let the other ones also comment, because in the doc trial we have used actually different depth to look at different properties. Who wants to start?
Shall I
Pablo.
start, Dimitris?
Yeah.
Okay.
Yes,
Yeah, in
sure.
terms of this, the depth will be really interesting to have in the models. Try to associate the moisture and the temperature of the soil depending on the depth. But it's already complex to predict something with just one layer of the soil, and maybe introducing more layers. Yeah, it will be a complex way to, let's say, to find out interesting patterns. And yeah, for this, as Dimitri says, it's complex to get sometimes information from the soils from one layer, and try to do global samplings with several layers is maybe it's a hard task. I don't know. Yeah, so it would be interesting to test. For sure, will be really interesting to have some projects where this another variable of the soil depth will be included in the models. It will be great to have this type of projects. Don't know, Dimitris, do you agree? What do you think about this depth?
No, I agree with you. I agree with the question, and I agree with your answer. It would be great to be able to do this. I mean, in terms of my use of experience, expertise, or interest, for me, it would be interesting to include that in our models. There are certain satellites, there are certain data sets derived from satellites that are able to penetrate, let's say, the soil a bit more, like radar or other types of sensors. And it would be very interesting to explore these sort of relationships, different soil depths, different microbiomes, different sensors that are able to explore these relationships. And it would be certainly interesting to explore them. I think for microservices, the Lucas dataset has, like, I think, a single sample depth for the biological sampling. So I mean, it was not possible to sort of expand the analysis towards that sort of front.
Okay, thank you. And then maybe also, since we're already talking about the depth effect, to Elena and Rainer, when we have basically measured, changes in soil moisture along the depth also that we have done too, we see that basically pretty quickly the drought becomes not so much of a big problem anymore. And you have shown basically how these properties vary quite a bit along the depth profile. So do we need to reconsider basically where we look for changes, because most studies still focus on the topsoil, the 20 centimeters? Do we miss something important when we study climate change effects on soils, when we only look at this topsoil?
Should I start, Rainer?
Yeah.
I think, it's quite important actually, because since water and nutrient cycling includes all these depth layers, we should sample deeper. And it's not that there is like a line and then nothing happens anymore. So it's all connected. So I think it's important that we start focusing also on deeper soil layers to include all this nutrient cycling, water cycling.
Yeah, I would agree with this comment. I think we see from the data that, of course, the activity at deeper layers is slower, lower microbial biomass, but the effects of drought seem to be stronger below 25 centimeter, because I think in this case plants try to get water from the deeper layers, and also this might affect nutrients and might also affect, in this case, we have a stronger carbon input in the deeper layers, and this is certainly also affecting microorganisms and microbial diversity.
Thank you both. Can the panelists actually see the questions as well?
Yeah.
Elena, can you type an answer to the question how you actually distinguish between rhizosphere and bulk soil quickly, how you have done this? So then because we only have basically 1 minute left, I would like to take the last one, which I think is of interest to many. It's about, the increased availability of AI tools and how we could use these in climate change and agriculture settings. So very general, broad question maybe to Pablo and Dimitris. What should researchers keep in mind when applying AI for such studies? I mean, thinking about error rates and, practicality and so forth.
Yeah, the first thing that we observe is it's useful to get some insights and, let's say, give some more concerns to the society in terms that we observe. For example, with the model that we develop under this project, where we compare different socioeconomic pathways, and for example, applying these models allow us to do first prediction of bacteria, and then how these bacteria impact on the functions, and then that the functions will be different depending on the socioeconomic pathway. So at the end, this can help, let's say, just to raise some concerns about the agriculture sector, and then this also introduce these models with more precise information, maybe about, focus only on productivity. Maybe they focus also other relevant functions. Maybe instead of speaking only about multifunctionality, splitting by ecosystem service, as was mentioned, and then and these models could help us to understand what could be the change in the future for each of the services.
Good. Thank you. Dimitris, anything to add? Quick response.
Very quickly, I would say that we are at the inflection point in AI, in machine learning. I mean, a random forest is different from an LLM, completely different. It's not the same quality of model. So that's one thing. But we are at an inflection point. Things are going to change very soon for all of us. What we use in our daily work is going to change very quickly. I mean, even for the models we're doing now, not just for summarizing, it's going to change very quickly. And we have to be sort of aware of that. We have to be able to understand what the models are doing, what they're not doing, because we're doing science. We're not summarizing to write an email. I mean, it's a different type of world. So as a first suggestion, start simple. That we if the question is what do we have to keep in mind, is to start simple. That there are some workforces in machine learning, like random forests, lasso regression. I mean, these are some tools that you can use first, and then you can sort of move on to more complex models. But everyone, I think, will move to more complex models eventually. Like they're more powerful, they're more.
Okay, thanks a lot. So with this, we're having only 2 minutes left. I would like to close the Q&A discussion, and I would basically go back to my own presentation, just have basically one slide left. Can you see my screen? Can someone quickly check?
Yeah, we see take home message.
Okay. So quickly, I try to, within 2 minutes, to summarize after like 4 years of very fun, but not always easy work in a really great team. So there, I also want to thank all of you, for contributing to this. What are my take-home messages, right? And I think, first of all, I think what we have seen are interesting first steps, how we can use indeed AI combined with, well, regional climate models to make climate predictions, Earth observation data from various sources, and molecular data on soil biodiversity, other soil data on functionality, to really predict or try to predict with a certain accuracy climate change impacts on soil biodiversity and ecosystem functions in agriculture. So I think there we have an interesting, innovative framework that obviously is a first step. We can build on this, but I think that is what I'm very proud of, that this came out of microservices. Second, I think a key insight is, and we have seen climate change or climate is a driver, but is not the primary driver. I think that maybe not came quite clear, but actually when we look, closer, it's primarily soil properties that drive a lot of these soil biodiversity shifts. And was also mentioned by Dimitris, right, that actually a lot of these soil characteristics are very important properties in the models in the end. So since local soil properties are so important, it highlights the need that we basically have to look for region-specific policies when we want to conserve soils for basically, climate change impacts. So because there's not one policy or no one strategy that will work across Europe, we need to consider this regional context to do it properly. Every region will respond differently. I think that Pablo has nicely shown climate action matters, right? In these prediction models, actually these low emission scenarios preserved soil biodiversity mediated ecosystem functions much better or consistently, right? So that basically reinforces that we need to act now. We need to have basically lower our climate change impact so that we can maintain these functions. So climate action matters. Even if it changes, if we can limit this change to a certain level, that already helps to still maintain a lot more of these important ecosystem functions. The fourth is, that's the second part, the legacy of the land use. I mean, despite we have seen similar magnitudes of these shifts in the different organic and conventional cropping systems in the stock trial, so that's only one trial, right? But one that is running for a long time, that is very different in terms of the organic matter content of the soil biodiversity and so forth. The magnitudes were the same, basically. But what we have found is that there were legacy effects. So certain plant growth promoting and stress tolerating traits, microbial traits, persisted also under the climate stress, which actually means that what we invest into soil health today, that will also then largely be maintained in the future. So we build resilience for tomorrow. And that's important to understand. It's not that once climate change hits, they all look the same and it doesn't matter. We can make a difference right now by choosing the right practices to increase soil health. And last but not least, I think that framework in microservices that offers actual evidence to basically shape land management climate policy so that we can protect biodiversity, protect our soil functions, and in the long run have a sustainable food system and therefore also food security. So the next step here is really also to distill a bit the information in microservices and make that available much broader to actually policymakers and other stakeholders. So these are my take-home messages. There will be many other interesting findings that we have seen in these 4 years. But with this, I would like to end that webinar, which also marks the end of microservices. So I would like to thank you very much for attending this webinar and for posing your questions, for listening. If you want to know more, please visit our website, microservices.bthz.ch, or drop me an email, or also if you have specific questions, obviously you will find all the partners also, I think, online. So with this, thank you very much for listening. Thanks a lot to all the panelists for the great presentations. And with this, microservices is over. Thank you very much and have a wonderful day.